Joining us from Bionano is CEO Erik Holmlin. Welcome, Erik.
Thank you, Sung-Ji. Thanks for having us.
Before I start with the questions, any introductory remarks you would like to make?
Well, I mean, we're obviously thrilled to be here and participate in the event, and we're big fans of you, by the way. You've been an excellent representative of the industry in the space, and so we look forward to this conversation. And we're very excited to talk about what optical genome mapping is and how it has the potential to really be the driver of the next big wave of big biology and genomics.
Fantastic. Thank you so much. So can we kick off with just the big picture, and then we'll peel the onion, so to speak. Could you talk about how Bionano Genomics platform, this optical imaging, optical mapping, I'm sorry, we're talking about a lot of different technologies right now. Optical mapping fits into the overall genetic analysis market, particularly compared to some of the next-gen sequencing platforms that are out there?
Sure. So optical genome mapping is a process that involves really, I think, 4 steps: isolation of extremely long, or what we call ultra-high molecular weight DNA, and we're talking about hundreds of thousands to millions of base pairs long. And we start with that type of starting material and then process it by labeling sequence-specific sites across these giant molecules, and then using the Sapphire system or instrument, which is a genome imager, to image those molecules while they're suspended in a nano-channel environment, so they're completely linearized. And from those images, our AI-based software analysis will extract the information from those images that tells us where those molecules originated in the sample, what chromosome they came from, what orientation they were in, and if there are multiple patterns, those multiplicities reveal the quantity of where those molecules are coming from. And so that could be a gene, a promoter region. And so that type of information, the genomic structure information, is readily obtained through this method. And we're uniquely capable of being able to discern that type of structural information. And it's because we're looking at these incredibly long molecules that span the repetitive sections of the genome. And that's a capability that is unmatched to our knowledge, to really look at single molecules that are so long and that span these repetitive regions.
Gotcha. And then could you maybe compare that to some of the next-gen sequencing platforms, such as, Illumina with their short-read sequencers, PacBio and Oxford Nanopore doing long-read sequencing? Kind of how, are these competitive platforms to each other in your view, or are they complementary?
Yeah, well, I mean, we see all sequencers as complementing optical genome mapping, and each process for those sequencers that you mentioned is a little bit different. Illumina and PacBio are using sequencing by synthesis, so they're building a copy of the molecule that they're seeking to sequence, one base pair at a time. Oxford Nanopore Technologies threads the molecules that they're seeking to sequence through nanopores, and so those are slightly different processes. But if you start with Illumina, the very first step of sample preparation for Illumina is to use a pretty traditional DNA extraction method that results in highly fragmented pieces of DNA, and that step really destroys the structural information to begin with, and then the sequencing is based on relatively short reads, or extremely short reads, 150 base pairs or so. And so that type of an approach is excellent at getting single base pair resolution. If you map those reads back to the reference genome, you'll be able to determine if there's been a single base change or a couple of base pairs have changed. But the problem with that type of an approach is that the genome itself is highly repetitive, and many chromosomes have sections that are relatively indistinguishable from other chromosomes. And so when you're mapping a read from some section of the chromosome, you may not know actually what chromosome it belongs on. And that information is obviously biologically important. That's why we have 23 pairs of chromosomes to begin with. And so if you actually don't know where the read is coming from in the genome, it's difficult to understand the biological significance. And clearly it's very challenging to determine whether there's been a change in genome structure. PacBio has sequencing, which is long reads. And by long, we're talking about, in the range of 10 to 20,000 base pairs, let's say. And the accuracy of those reads has improved dramatically over time. And so it's really an amazing platform for getting long reads that are highly accurate. But those reads have limitations in terms of looking at structural changes that are in the range of what's clinically relevant, hundreds of thousands of base pairs, millions of base pairs in some cases. And so, the Sapphire system with its molecule lengths that are hundreds of thousands of base pairs long really overcome that limitation. Oxford Nanopore Technologies is somewhere in between. They're not doing a sequencing by synthesis approach, so they don't have the limitations of those reads, but the molecule lengths that are threaded through their nanopores tend to be smaller in size, and then there are accuracy issues, again, with the repetitive regions. Now, the way that the two technologies, sequencing and mapping, are complementary is that with a sequencing solution like Illumina, it's possible to determine with extremely high accuracy whether there are single nucleotide changes, single nucleotide variants across the genome. And that type of information can be biologically relevant, clinically relevant. And then coupling that with optical mapping, which reveals all of the structural variations, 500 base pairs and bigger, with high sensitivity, low false positives, it becomes possible to have comprehensive lists of genome variation, which is incredibly powerful in discovery research and has applications in clinical diagnostics as well.
Great. So Erik, could we talk about, just in terms of genomic variation, you talked about there are reasons why Illumina platform or PacBio platform, Illumina especially, that really has taken off. They're doing short read sequencing. And as we look at genomic variations, a lot of common ones, like you talked about, single nucleotide polymorphisms, maybe short insertions, deletions, et cetera. So could you talk about kind of, structural analysis? Obviously that's an important component, and you guys are actually showing it is definitely more relevant and important in understanding the different variations and et cetera in drug development, discovery development, all the way to diagnostic development and whatnot. But how should we think about, as you look at just where the science is today, in terms of how important, or what percentage, I guess, is there a way to quantify what aspect of the variation analysis falls into structural variations that would require a technology platform like yours?
Sure. If we look across clinical diagnostics today, virtually every patient who is suspected of and being evaluated for some form of a genetic disorder is having a structural variation analysis conducted. That's what the medical guidelines recommend. ACOG in neonatal and prenatal testing, Academy of Pediatrics. So these ACMG, these guideline-setting agencies prescribe the use of chromosomal microarrays, which is probably the best structural variation analysis tool that's used routinely in the clinic today to look for structural variations. The same is true in cancer. So karyotyping and FISH, these are tools that look for structural variation. And if you were suspected of or being evaluated for a hematologic malignancy like a leukemia or a lymphoma or solid tumor, there's going to be structural variation analysis. So SV analysis is the standard of care today. We know it as anatomic pathology, molecular pathology, cytogenomics, cytogenetics. We don't always talk about it in the variant terms. Those are actually kind of like sequencing terms, but it's very common. And being somebody who's in the structural variation field, I think it's more common. But that's something to actually be measured by somebody who wants to quantify that. But it's incredibly common. So it's the standard. What's very different, totally unique, is that if you think about the first 20 years of this century, in genomics, the progress has been driven by tools that were basically industrial scale. First, microarrays, which drove a lot of gene expression analysis, SNP association studies, and they found their ways into clinical applications a lot because of how useful they were, right? Relatively easy. Illumina has done an amazing job at making the sequencing accurate, then faster and cheaper. If we look at structural variation analysis, what do you have? What's available to you in cytogenetics? You really have 3, 4, 5 different platforms you need to do a complete structural variation analysis: karyotyping, chromosomal microarrays, fluorescence in situ hybridization FISH, southern blot, PCR. So 5 different tools are required to do a complete structural variation analysis workup. And so we don't hear as much about structural variation because there isn't an industrial scale tool to do it, except, of course, now we have Saphyr. So Saphyr has the ability to make structural variation analysis as accessible, as fast, and as accurate as Illumina has made sequencing. Think about Sanger sequencing. I would never be able to be used at the scale that NGS is used at today, but now it is. And so the Saphyr system with optical genome mapping really is to structural variation analysis what NGS was to Sanger sequencing. And so I think you're going to see a lot more use of it in discovery research. Think about the rare disease companies like the FSHD companies and how empowering it's going to be for them to search for those repeat expansions and contractions in much broader patient populations. But FSHD is just the tip of the iceberg. There are many, many more rare diseases that Saphyr can help uncover the driver mutations for. And then already today, cancer is a disease of structural variation. So Saphyr will play a prominent role in that.
Fantastic. So could we talk a little bit more about, you touched on discovery research, obviously applications there. Discovery research, there's still a lot of short read sequencing and also increasingly long read sequencing going into that. Just how do you see that market for you guys? And I guess along with that, just if you could talk about the total addressable market. I think you guys have done an analysis of that. So how do you look at discovery research for every 10 Illumina sequencers with, a large lab looking at, a genomics lab, is it typical for them to acquire one Saphyr platform? Kind of trying to better understand.
Yeah, I mean, I think at a steady state, it's reasonable. I mean, to think of a structural variation analysis being useful on a one-to-one basis with a sequencing analysis. Now, there is going to be biological questions where the answer is SNP-based, and so you'll use sequencing to go for it, but the opposite is true. So that probably levels out. I think it's reasonable to think of every sample that's being sequenced getting a structural variation analysis alongside with that. When it comes to like what's the right ratio of equipment, you have to boil down what is the throughput and so forth. But we've done some analysis, and at one point in time, there were about 7,000 high throughput sequencers out there. And our definition of high throughput is if the sequencer is capable of doing 1 human genome per day. 7,000 of those out there. Now, that number is actually shrinking a little bit because NovaSeq's capacity is higher than HiSeq. So as there's a conversion from HiSeq into NovaSeq, you see the number coming down. But think about it as the capacity, of these genomes. We see it's completely reasonable that there would be a 1-to-1 ratio of mappers to sequencers out there. And so that's a significant opportunity on the discovery research side. And then the complement is in cytogenetics, where there's not a lot of overlap with sequencing because, as we've been talking about, cytogenetics is about chromosomal structure analysis, and sequencers are not generally that useful at it. There is overlap within molecular diagnostics and molecular pathology, but generally there's about 2,500 cytogenetics labs, pathology labs around the world that would be adopting this type of technology. So altogether, we generally look at the total available market for system placements as being around 10,000 Saphyr systems.
Okay, gotcha. That's inclusive both cytogenetics and discovery research.
Cyto and discovery.
Fantastic. And then could you talk about maybe, or could you give us some case studies of where, where the Saphyr platform is able to, detect things that other platforms just cannot? I think you use, in some of your presentations, the Duchenne muscular dystrophy diagnosis. There are some head-to-head comparisons now emerging in terms of looking at your platform versus some of the other platforms out there. So we'd love to hear just actual,
Yeah.
real world examples
Yeah.
of where you're able to see things that other platforms cannot.
Yeah. So I think this is one of the really compelling aspects of Saphyr and optical genome mapping. Again, is that it's complementary. So if there's a SNP at the heart of the biology, Saphyr won't pick it up, sequencing will. So use sequencing. But if you're really trying to dig into the biology or a diagnosis in the case of an otherwise undiagnosed genetic disorder, Saphyr is the perfect platform to be using. And you mentioned the Duchenne muscular dystrophy example. There are several of them out there now, but the one case that's commonly talked about is a young boy, 9 years old, clearly exhibiting symptoms of muscular dystrophy. Like on clinical presentation, it's clear that it's muscular dystrophy, but a definitive molecular diagnosis is critical because patients and their families can end the diagnostic odyssey based on that. Payers are keen to have that completed because they too can stop paying for the diagnostic odyssey, which can last for years and be very expensive. And in this particular case, this boy had seen a number of physicians and specialists over the course of his life. And fortunately, he ended up in a program that had his sample at Children's National Hospital in Washington, D.C., in the group of Dr. Erik there, who is a Saphyr user and a supporter of Bionano and a great collaborator. And they ran that boy's sample in a single run and immediately found a large segment of that boy's chromosome 15, 410,000 base pairs that had been duplicated. So chromosome 15 was normal, but 410,000 base pairs of it were copied and inserted into the boy's X chromosome. And it just so happens that those base pairs were inserted right in the middle of the DMD gene. So basically severely disrupted the normal function of that gene and therefore explained the boy's disease. Why wasn't that picked up with sequencing? Well, it's very complex. In this case, the sequence hasn't changed. It's a section of chromosome 15 that's showing up somewhere else. So when you go in with these tiny reads in a completely shredded up genome, that becomes difficult to discern. Certainly it's the chromosome 15 sequence. It's in a different location. The structure has changed. The sequence hasn't changed. That's why Bionano picks it up. And of course, as you know, in Duchenne muscular dystrophy, there are therapies that are available now. And so these types of findings are incredibly important. And there was a study that was published by UC San Francisco in collaboration with Oakland Children's Hospital, where they looked at a population, 50 children who were undiagnosed, and they used the Saphyr to analyze them, and they were able to diagnose an additional 18% of those patients. So when you think about the typical performance of a genetic testing lab using the traditional methods today, 30 to 50% of patients, they're able to find a definitive pathogenic finding in. And so by adding an additional 18% or diagnosing 18% of the otherwise undiagnosed patient population, this really moves the needle. But what's also powerful about Saphyr is it can take the place of the initial traditional method. So instead of 5 techniques, you use one and you get a higher diagnostic yield. So that's one example. And there are many others where sequencing either wasn't used in a whole genome sense because of cost, or panels were used, and so the Saphyr system is completely unbiased and looks on a whole genome basis and has a much higher chance of revealing some of these driver events.
Gotcha. So it sounds like there are, a lot of obviously clinical applications, translational research applications, et cetera. Do you guys have any plans, or have you talked about any plans to really get into the clinical market? And if you, or they're already, I guess, available there, but, just love to hear your strategy in terms of, would you pursue regulatory clearance for the platform, use it as a more distributed platform? Would you provide it as a service? And you acquired the Neogen, right? And we can talk about that a little bit later, but love to hear your strategy there.
Yeah, definitely. So yes, actually our current strategy, going to market now, Saphyr is a research use only platform, but clinical labs can adopt it, validate an LDT on it, and then use that LDT for clinical applications. That would be their proprietary LDT, and that's a common way in which these technologies are adopted in the clinic. And that's our current target market. And what we believe is that when the Saphyr system is broadly used in these clinical applications, that the use of it in discovery research will start to accelerate. Because imagine if you make a clinical discovery that's biologically significant, and then you want those patients to be able to get tested for it in the clinic, you need the system available clinically. So we're going after this, these labs that will develop LDTs. When you talk about Lineagen, the thinking in bringing that in is to leverage their channel, so direct to patients. We have patients that are coming in. We can use those samples in research to develop assays on Saphyr. We're now running a series of clinical trials that are getting designed and kicked off, 4 of them, one in prenatal genetic testing, one in postnatal genetic testing, in hematologic malignancies, so leukemia and lymphoma, and then solid tumors. And those trials are designed to build the database and collection of laboratory developed tests and proprietary laboratory analysis, or PLA codes, that will promote a reimbursement of Saphyr by third-party payers. And so this Lineagen acquisition brings in all of those capabilities, a lot of samples, a lot of proprietary databases, patient connections, and it really accelerates our path to the clinic. It also adds revenues, so they're providing cutting-edge diagnostics to pediatric developmental specialists, and who are testing kids for autism spectrum disorder, for example. And so, those revenues are adding to the Bionano revenues around Saphyr sales, while we clear the path to adoption clinically. And I want to make one last comment, and that's that this path to adoption in cytogenetics is really taking off in Europe. And the difference there is that these are largely single-payer healthcare systems, so they don't have the pressure from third-party payers that we have here in the U.S. And so the Lineagen acquisition and these clinical trials are going to clear the path to that reimbursement, and we expect a lot of that to unfold over 2021.
Great. Maybe going back to the install base, or the kind of the addressable market of roughly 10,000 systems. I think when Illumina first started, I don't think they could have imagined that they would exceed that, and they're already exceeding that, right? I think their latest is maybe over 12,000. And so could you talk about, are there, I know it's early on and you have a lot on your plate, getting to that 10,000, but are there other opportunities beyond cytogenetics, beyond kind of the basic research, other applications in your view that this, platform could enter in the future?
Well, yes, for sure. I mean, and I'm seeing the cues about our time remaining here. For sure, a whole area of utility in agrigenomics, outside, non-human, but not just agri. Think about mouse and other model genomes And so, this is an area of significant utility, and so there are many genomic applications. But what we see and what we're focused on in terms of that, really huge future potential, and of course, we're seeing others like Illumina follow this path, and that is that there's a lot of content development that can happen for us, and we can monetize that. So different services offerings, but different novel genomics-based tests that are structural variation oriented, and so we'll be investing in that content development, and we believe that can be a source of future revenues and future growth for the company that are based on content that's unique to